So here we have a picture of some fish in the water. And in the water there are dissolved nutrients and oxygen that the fish use to exist. In this picture we have a picture that looks somewhat similar, only this is on the cellular level, and it shows basically the fluid system outside the cell and the fluid system inside the cell. These are both aqueous in nature, means that the main component of this is water, and there are things dissolved in that water. Now, what kind of motivates our discussion here is the need for substances of various types to move into and out of the cell. So what we want to do is look at how those things behave in those aqueous solutions, in the water solutions, so we can understand better how they might move into and out of the cell. So here's another image of that cell wall, which is that phospholipid bilayer, and we'll get into talking a little bit more about how that functions in the process of moving things into and out of the cell. We know that the fluid part inside the cell we refer to as the cytoplasm, and the fluid part outside the cell is the extracellular fluid. And we can see all the different, there's the large purple things are proteins that will help facilitate the movement of substances into and out of the cell, and we'll learn a little bit more about those as well. If we take a closer look at that phospholipid bilayer, what we have at one end of each of those molecules that come together to form that phospholipid bilayer, so that round ball part in here that you see in the animations is this part right here. And this is a polar part of that molecule. So this is the part that would have a positive and negative end and would be soluble in water. So it would be hydrophilic. It would be a water-liking part. And then that tail part that's on there is the nonpolar part. So recall, talking about polar and nonpolar molecules. So that tail part, which is in here and in here, will be the nonpolar part. So that's the hydrophobic part, the part that doesn't necessarily like water. Now remember, in all these diagrams that oftentimes to help you understand the function and what's happening on the cellular level better, you're left to kind of imagine what the atoms and the bonds and things look like in the animation. So always kind of keep in the back of your mind that although you see these interesting caricatures and things here, that the fundamental nature of those is that they are atoms or atoms that are bonded together forming molecules, and oftentimes those molecules are very large and very complex. All right, so the main question we have then is, how do we move stuff into and out of the cell, and what stuff is there that we want to move into and out of the cell? So we probably have some nutrients that we need to move into the cell, energy molecules, perhaps some proteins that may move into or out of the cells. Water will be something that would move into and out of the cells. The wastes that are produced within the cell would need to move out of the cell. There are some gases that we might need, carbon dioxide, oxygen, that would be either produced or needed in some of the processes inside the cell that we need to move across those membranes as well. So, and there may be other things too that that we come across that need to move, both into and out of the cell. The key to understanding how these things will behave in response to the phospholipid bilayer is looking at whether they are large molecules or small molecules, and whether they are polar or nonpolar or ionic in nature. Those are the questions we're going to want to ask ourselves in terms of trying to figure out how these things would be able to move into and out of the cell. So let's take a look at some different molecular pictures of each of these types of things. All right, so here we have an ionic substance mixed in with water. And so as you see in the picture, the water molecules are moving around this ionic substance. And because water molecules are polar, so they have a positive and negative end, they will break apart that ionic substance. And we'll pause this to take a look at a couple of those different species that are being formed here. Right here. All right, so if we look at what we've got here, here we have one ion. So if we imagine this being perhaps a sodium chloride, this being the negative chloride ion in the green here, you can see that the water molecule is surrounding that with the hydrogen side toward it. The hydrogen side of the water molecule is the polar side, and so it's surrounding that negative ion. The small purple dot then, and just below that, here, that one is going to be the sodium ion, and you can see that the red or the oxygen side of the water molecule is basically surrounding that part, and the oxygen being the negative part of that molecule. So this ionic substance, when it gets dissolved in water, it's going to break apart into the positive and negative ions, and it's surrounded by the water. This happens fairly easily and facilitates the dissolving of this, and so you have these negative particles then dissolved in the water. Here's a better picture of that, where we can see, again, the negative ion here surrounded by the water molecules, the hydrogen end pointing toward that, the hydrogen end being the positive part of the water molecule. Again, we can see that over here. And the positive ion, the cation, being surrounded by the water molecules. But in this case, the oxygen side would be closer to the positive part there. So what happens with larger molecules being mixed in with the water? So first we'll take a look at sucrose dissolved in water. Sucrose is going to have polar parts to the water molecule. So sucrose is definitely a polar type of molecule. Sucrose we know dissolves in water. So we'll take a look at what that image is. So here's an animation of the sucrose. I'm not sure exactly how well this is going to work on your computer, but you have the sucrose molecules then that are able to be surrounded by the water molecules. Notice in some places you have the oxygen end toward the sucrose, and other parts of it you have the hydrogen. But because sucrose is a polar molecule, it's able to dissolve in and interact with the water molecules. Now, if I do the same thing with oil mixing with water, I get a slightly different picture of what happens here. So here are the water molecules, and I'm going to bring the oil molecule in here. The oil molecule is nonpolar. So there's no part of the water molecule that will interact or try to bond to the oil molecule. As a matter of fact, the water molecules really would rather attract to themselves because they're polar. So what this is going to do is it's going to tend to squeeze that oil molecule out so that the water molecules can be together. Again, because of the polar nature of them, they're positive and negative, so they're going to attract each other. So what happens then is that the oil will separate from the water, which is what we see happening if we do mix oil and water. You see this with salad dressing and things like that. So here I have a picture of what we would imagine on the macroscopic level, where we have the water down at the bottom and the oil on top of it again, because they do not mix. So the oil is more of a hydrophobic substance. So basically our discussion boils down to what type of substances do we need to move into and out of the cell through the phospholipid bilayer. There's two general categories: those that are hydrophilic, which are dissolve easily in water. These are your polar molecules and your ions. And those that are hydrophobic, the nonpolar types of molecules. So in the hydrophilic section, the simplest things are our ions. So we've got our potassium ions or sodium ions, for example, chloride ions. So positive and negative ions that will dissolve usually very well in water. Then we have our small polar molecules, such as water itself, would be an example. Ethanol, so C2H6OH, methanol, urea. So there are other small molecules that are polar in nature. Again, polar molecules being ones in which the electrons are shifted toward one side of the molecule due to difference in electronegativities of the atoms as well as the shape of the molecule. Okay, then we have larger polar molecules such as glucose and sucrose, which again are polar in nature. And then we have our hydrophobic, our non-polar molecules like oil. But they can also be small. So we consider oxygen as being a non-polar molecule. CO2, nitrogen in the air, other larger molecules such as steroids and things like that that we might need to move into and out of the cell would be hydrophobic due to the non-polar nature of their molecules. So the next thing we're going to want to look at is the process by which these things move into and out of the cell through the phospholipid bilayer, and that will be the subject of our next video.